Thank you for that introduction, Dean, and good morning, everyone. I can't believe it's been 3 years since we conducted the QFever survey at the Rehabilitation Conference in 2018 that some of you may have even participated in. And today I'm really pleased to be here to share the findings of this research with you. So 2 manuscripts emerged from this research. One was on QFever and one was on Rickettsia. So first I'll present the findings of the QFever study and then I'll move on to the Rickettsia one. So what is QFever? Well, QFever is a disease transmitted from animals to humans caused by the bacterium Coxellia burnetti. Now, Coxellia burnetti is an intracellular bacterium in that it needs to grow in a host so it can replicate as well. So over here we've got a picture of a vero cell, and these Coxellia burnetti grows in these special compartments within the cell called a vacuole. Now, because of these intracellular growth requirements, the organism can't be grown using traditional culture techniques, and it requires special physical containment level 3 facilities to be grown because it's so infectious. So it is a little bit tricky to work with. So outside the host, Coxellia burnetti exists as a stable spore-like form, which can remain viable in the environment for at least 12 months. So Coxellia burnetti is found worldwide except for New Zealand and Antarctica. However, it was first described in 1935 in Australian abattoir workers by this fellow up here, Edward Derrick. And the cause of the illness was unknown and it was called QFever. So that's where the Q comes from, in case you ever wondered. So although QFever has been a notifiable disease in all states and territories since 1977, and although it's been found throughout Australia, the majority of notifications are from New South Wales and Queensland, and these 2 states accounted for 85% of notifications in 2020. So the main way of contracting QFever is by inhaling aerosols which are contaminated with the bacterium. And following infection, the clinical presentation of QFever is quite variable and complicated. So approximately 60% of cases are asymptomatic, and people with symptomatic infection most commonly present with a flu-like illness that may occur in conjunction with complications such as hepatitis and pneumonia. Chronic QFever infection can result in QFever fatigue syndrome or manifest as a persistent focalized infection such as endocarditis. However, regardless of whether the infection source is symptomatic or asymptomatic, by this diagram you can see that both can lead to serious long-term health consequences in humans. So although Coxellia burnetti infection has been identified in both wild and domestic animal species, cattle, sheep and goats are regarded as the main sources of infection for humans. And this is reflected in the QFever notification data with a high proportion of notifications from the meat and livestock industries. So infected animals can shed their organism in large numbers, particularly in birth products, but it can also be shed in urine, feces and milk of these infected animals. So Coxellia burnetti has also been identified in over 40 species of ticks. However, transmission to humans by ticks is rarely reported and poorly understood. So while livestock are without doubt the main sources of human infection, over the past decade or so, there's been an increase in notifications where these traditional exposure sources could not be identified. And there's a growing body of evidence to suggest that kangaroos and wallabies are potential sources of infection for humans. So QFever has been reported in people working in outdoor environments inhabited by kangaroos, in individuals mowing grass heavily contaminated with kangaroo feces, and contact with joeys and ongoing exposure to kangaroo and wallaby carcasses involved in road accidents have also been considered as potential sources of exposure for humans. So what do we know about macropods and QFever? So Coxellia burnetti has been isolated from the blood and ticks of infected kangaroos. And seroprevalence studies for Coxellia burnetti antibodies in kangaroos in Western Australia and Queensland have reported 25 to 33% seroprevalence in these animals. The DNA of Coxellia burnetti has also been detected in kangaroo feces and in pet meat containing kangaroo. And while these studies provide evidence that kangaroos may become infected, so far the evidence for macropods as a source of Q fever in humans is largely circumstantial. So that's where I come in. So my research question is essentially, are Australian wildlife species, with a particular emphasis on kangaroos, a source of Q fever for humans? So to help us understand the connection between kangaroos or wildlife and Q fever, I conducted the serological survey at the Rehabilitation Conference in 2018. Now, the aims of this research were to determine the extent of exposure to Coxellia burnetti, so look for Q fever antibodies in rehabilitators who care for Australian mammals. I also wanted to assess the level of Q fever disease and see how many participants were vaccinated. And once I did this, I wanted to look for potential risk factors for exposure to Q fever, for Q fever disease, and for Q fever vaccination. And I'll go into a little bit more detail about these risk factors a bit later on. So this was a nationwide serological survey conducted at the Australian Wildlife Rehabilitation Conference in 2018. And to be eligible to participate, you had to be over the age of 18 years of age and care for Australian mammals. So participants completed a questionnaire to go with their blood sample, and the serum was sent down to the Australian Rickettsia reference laboratory, where it was tested for Coxellia burnetti antibodies using immunofluorescence, which is the gold standard diagnostic technique for Q fever. So for the analysis, I was interested in the number of participants who reported having had Q fever disease. And I was also interested in the number of people who were seropositive, that is the number of participants who had evidence of exposure to Coxellia burnetti as evidenced by antibodies in their blood. And I was also interested in the number of participants who had been vaccinated. Then I performed a statistical analysis to try and identify risk factors for Coxellia burnetti exposure and Q fever vaccination in this group. So what I wanted to know was, what is it that you might do or make it more likely for you to be exposed to Q fever, and what type of people are more likely to be vaccinated? So a quick look at the demographics of the cohort. So the majority of the cohort were female. They had a wide age range from 21 to 79 years with a mean age of 51 years. Looking at the percentage of respondents from each state, we can see that New South Wales ACT had the largest number of participants, which is, I guess, not surprising considering it was the host state, and this was followed by Western Australia and Victoria. 59% of participants reported occupational contact with animals, 61% rehabilitated macropods, and 44% reported being bitten by a tick. And we can see by this graph that the most frequently rehabilitated animals were possums and gliders, followed by kangaroos, wallabies, flying foxes, echidnas, wombats, bandicoots, wallaroos, koalas, and pademelons. So let's take a look at the results. So approximately 350 Australian wildlife rehabilitators attended the conference, and almost half of these delegates participated in the study, which is absolutely fantastic. We were really, really stoked with this. So thank you all for participating. We actually had to remove 5 from the data set as they were unsure of their vaccination status, and this left us with 160 participants in the final data set. So first I grouped the data set according to whether they were vaccinated or unvaccinated. So let's have a look at the unvaccinated group. So of the unvaccinated participants, 3 reported having had Q fever disease, which is significantly higher than what we see in the general Australian population. Turning our attention now to the exposure, so looking for Coxellia burnetti antibodies in the blood of the rehabilitators. So 9 of the unvaccinated participants were seropositive for Coxellia burnetti. And this is almost twice as high as what we find in healthy Australian blood donors. Now, turning our attention to the vaccinated group. Now, despite vaccination being recommended for this group in the Australian Immunisation Handbook, only 13 of the 160 participants were vaccinated against Q fever. So next I performed a risk factor analysis for exposure to Q fever and vaccination. So let's look at the risk factor analysis for Q fever exposure. So I wanted to know what it was that these participants might do to make them more likely to be exposed to Coxellia. So I looked at various factors that I extracted from the questionnaire that they answered that went with their blood sample. So I looked at demographic characteristics such as state of residence, where they cared for the animals, their age, the number of household members caring for wildlife, and education. I also looked at the years caring for Australian mammals, how animals were housed, and the number of animals cared for per year. I explored occupational contact with animals, looked at biosecurity practices when handling animals and cleaning enclosures, and whether the participants had been bitten by a tick. I looked at being present at births, non-human births, and if participants handled joeys. And on the animal side of things, I looked for correlations with traditional sources such as cattle, sheep, and goats, as well as domestic animals, kangaroos, and wildlife. And lastly, I looked at the frequency of caring for macropods over the past year and over the rehabilitation career of the carers. However, despite looking at all these risk factors, I was unable to find any that were associated with exposure to Coxellia burnetti. So I did a similar analysis to try and determine what type of people are more likely to be vaccinated. And I found 3 factors associated with vaccination. So what this table shows is that rehabilitators who were from Queensland were twice as likely to be vaccinated against QFever compared to rehabilitators from New South Wales, and 3.5 times more likely to be vaccinated than rehabilitators residing in other Australian states and territories. Rehabilitators who cared for between 31 and 100 animals per year were 2.8 times more likely to be vaccinated against QFever compared to those who rehabilitated less than 30 animals per year. And finally, rehabilitators who reported occupational contact with cattle, sheep, or goats, so the traditional sources of QFever, were 8 times more likely to have received the QFever vaccine than people who reported no animal contact or contact with other animals as part of their occupation. So in conclusion, given that Australian wildlife rehabilitators in this study reported a higher level of QFever disease and were almost twice as likely to be exposed to Coxellia, we concluded that these wildlife rehabilitators are at increased risk of contracting QFever compared to the general Australian population. However, the activities associated with this increased risk of exposure were unclear. However, what was evident was Australian wildlife rehabilitators are exposed to a wide variety of animal species and environments which are potentially contaminated with Coxellia burnetti. And irrespective of the exposure source, QFever is a disease that should be avoided at all costs. Therefore, as per national guidelines recommended in the Australian Immunisation Handbook, vaccination is recommended for rehabilitators of Australian wildlife. Okay, so moving on to the Rickettsia study now. So this was a retrospective study in that it was not planned at the time of the conference to be done with the QFever sero survey. And I contacted people who opted to receive their QFever results and asked them for permission to test their blood samples for Rickettsia. So what are Rickettsia? Some of you may not have heard of Rickettsia, so we'll just give you a little bit of background here. So Rickettsia are intracellular bacteria transmitted by arthropod vectors, and by that I mean ticks, fleas, lice, and mites. And so, yeah, they're similar to Coxellia in that they need to grow inside a host cell. And what we see over here is Rickettsia growing in tick hemolymph cells. So these are equivalent to human blood cells. So animal species are the reservoir hosts in which they grow and replicate, and humans are generally accidental hosts. So these Rickettsia actually parasitize various human cell types accidentally. They're classified into 3 main groups. So we've got spotted fever group, the typhus group, and the scrub typhus group. So people become infected with Rickettsia through bites from these arthropod vectors during feeding in the case of ticks and mites. And these arthropod vectors also shed large number of Rickettsia in their feces while they're feeding. In the case of fleas and lice, the contamination of the bite site usually occurs during feeding from feces. Or it can also enter via abraded skin or via conjunctiva. So what are the symptoms of rickettsial disease? Well, I guess these are the non-specific symptoms. So you get fever, headache and chills. However, the most specific symptoms of rickettsia are the formation of this eschar. So this is typically what is found at the bite site of a tick or a mite. So it's a piece of dead tissue, and it's a telltale sign that you've been bitten by a tick. A lot of rickettsial infections also display a rash, a characteristic rash. So there are several rickettsia of clinical importance in Australia, and let's have a look at the spotted fever group first. So this table on this, we've got the rickettsia species, we've got the disease it causes, the distribution of the disease, the arthropod vector and species, and the vertebrate host. So we'll start with Rickettsia australis. So Rickettsia australis causes Queensland tick typhus, and it was the first rickettsial disease described in Australia, and it's found along the eastern seaboard of the country. It's transmitted by 3 known tick species, and the vertebrate host are native rats and bandicoots. So we're looking now at Rickettsia honii, which causes Flinders Island spotted fever, and it was first described in Flinders Island in Bass Strait, and cases have since been found in South Australia and Western Australia. And this is transmitted by the southern reptile tick, whose vertebrate host is the blue-tongued lizards or snakes. Another closely related species is Rickettsia honii subspecies Marmonii, and it causes Australian spotted fever, cases of which have been found in far north Queensland, Tasmania and South Australia. Now, the last member of the spotted fever group we want to talk about is Rickettsia felis, which is a little bit of an outlier because unlike these other 3 that are transmitted by ticks, it's actually transmitted by a flea, in particular the cat flea. And Rickettsia felis causes cat flea typhus. So moving on to the typhus group now. So Rickettsia typhi causes murine typhus, and it's transmitted by a flea also, and it's been found throughout Australia. So Orientia susugamushi, that's a mouthful, causes scrub typhus, and it's found in tropical Australia. And it's transmitted by the larval form of the mite Leptotrombidium deliens. So the aims of this research were to determine the level of exposure of Rickettsia species in Australian wildlife rehabilitators and investigate the association between exposure to Rickettsia and risk factors for exposure. And I also wanted to try and identify any current infections in the cohort using PCR to detect Rickettsia DNA in the blood and serum samples of participants. So the methodology was similar to the QFever study, but this time the serum samples were tested for exposure to Rickettsia species. And I also extracted DNA from the whole blood and serum of the participants and used a PCR designed to detect rickettsial DNA in these samples. I then performed, so I was interested to see how many people had evidence of exposure to Rickettsia species, so antibodies within their blood. I wanted to know how many people had recently been infected, and then I wanted to perform a statistical analysis to identify potential risk factors for exposure to Rickettsia. However, the questionnaire that I used to extract the risk factors from was designed for the QFever study, so it didn't contain any questions regarding the clinical history or the symptoms of rickettsial disease and questions pertaining to activities that it would expose participants to ticks. So let's take a little look at what I found. So overall, 122 participants responded to the invitation to participate in the study and consented to having their blood tested for evidence of exposure to Rickettsia species. And of these, 22% tested positive or seropositive, so they had antibodies to Rickettsia species in their blood. And none of the 122 participants had evidence of current infection, so I was unable to detect Rickettsia DNA in the blood of any of the participants. So we've got 27 people who were seropositive, so let's have a look at how they were classified. So the serum of these participants was titrated against the spotted fever group species, and I found that 4 of the 27 participants were positive for Rickettsia australis. So remember, Rickettsia australis was the one that caused Queensland tick typhus, and obviously it's a tick-transmitted Rickettsia. One of the 27 was classified as a Rickettsia honii infection, and this was the one that caused Flinders Island spotted fever, which is also tick transmitted. And one of the participants was classified as a Rickettsia felis infection, and remember, the Rickettsia felis was the one that was transmitted by the flea. Okay, so 18 of the 27, so we've got 5 here that are tick transmitted, and a further 18 of the 27 were classified as indeterminate Rickettsia australis and Rickettsia honii infections. So what that means is that I was unable to determine which species was responsible for the infection because of the cross-reactivity between the rickettsial antigens. But what's really important here is that these 2 rickettsial species are tick transmitted. And interestingly, despite the fact that these 23 participants had been infected with Rickettsia species that are known to be tick transmitted, only 11 out of the 23 reported having been bitten by a tick. And this indicates that a lot of tick bites go unnoticed. When I titrated the blood samples against the typhus group and the scrub typhus group, no participants were positive for Rickettsia typhi or Orientia susugumushi. So how does this compare to what has been reported in the scientific literature? Well, a Western Australian study looked at exposure to tick-transmitted Rickettsia in regainers. So regaining is a sport similar to orienteering, where participants spend up to 8 hours in the bush and are exposed to ticks. And this study found that regainers had 23% of the regainers had been exposed to tick-transmitted Rickettsia, compared to 2% in the control group with no known tick exposure. Another study looked at exposure of in chronically ill patients from New South Wales and Queensland, and found that approximately 40% of ill patients had been exposed to tick-transmitted Rickettsia, compared to 6% in the control group. Now, it's important to point out here that these samples here, this testing here was done on serum samples from ill people, and this study here was done on healthy people. So when we're comparing my results, we can see that it more closely aligns, the population more closely aligns to the regainer study, and the actual seroprevalence that I found, which is 22%, is very similar to what was found in this study as well, which is much higher than what is found in the general population of healthy people. So in conclusion, we found that 22% of Australian wildlife rehabilitators had antibodies to Rickettsia species, and the majority of the infections were from Rickettsial species known to be transmitted by ticks. However, less than half of the exposed participants recalled being bitten by a tick. Unable to find any risk factors associated with the tick exposure. However, the health consequences of rickettsial infections can be severe, so it's really important for rehabilitators of Australian wildlife to be aware of ticks. And so this little diagram on the side shows a finger, and it shows the different life stages of a tick. So we've got the the larva, the nymph, and the female and the male adult. So you can see that when you're looking for a tick, they may be very small, so you have to look very hard. So this is sort of what you need to be looking for. So things that you can do to avoid being bitten by a tick include wearing long sleeves and pants when you're out in tick-infested areas. Wearing light-coloured clothing helps you to be able to see ticks more clearly. Applying insect repellent to deter ticks, and also is really important to check for ticks regularly on animals and after being outdoors. And I just found this diagram here on the Centre for Disease Control website. It shows you where you can actually, the best places to check for ticks, where they like to lodge, And it's also got information about how to remove ticks, because it's really important that ticks are removed correctly. Also, it's really important to be aware of the signs of rickettsial illness. So while these symptoms up here might be very non-specific, the eschar and the rash are really important telltale signs that you may have been bitten by a tick. And if you do remove a tick, it's a really good idea to keep it and take it with you to your doctor. There's a fact sheet available on Rickettsia on the New South Wales Health website. So if you just Google New South Wales Health Rickettsia fact sheet, you'll be able to download a fact sheet on Rickettsia. And lastly, I'd like to acknowledge the Australian Wildlife Rehabilitators and the Conference Organising Committee in 2018. I'd like to thank Liana Bonner for helping out at the conference, and all my wonderful supervisors at the University of Sydney, collaborators, the funding bodies, Marie Bashir Institute and the NHMRC, and the collaborators from the Australian Rickettsia Reference Laboratory. Thank you. I'll take some questions, but first of all, I just wanted to put this slide up here. So there's a Foundation for National Parks and Wildlife are currently having a QFever vaccination grants available for licensed groups, and Samantha McCann actually asked me to put this slide up. So if you're interested in applying for a grant for vaccination, you can contact Samantha, or there's more information available on the HEROES website here. Thank you.